Good afternoon, everyone. First of all, I'd like to thank Tracy and John for the invitation to present my work here and for putting together this exciting day of science at this beautiful venue for us all to enjoy. So as Mark mentioned, today I'll be talking to you about aberrant B-cell responses in chronic obstructive pulmonary disorder, commonly known as COPD. Not that this audience needs an introduction to B cells, but I would like to quickly highlight the many diverse roles that B cells play in the immune response against pathogens. So in addition to their unique ability to produce antibodies, B cells are also important antigen-presenting cells that present peptide antigens to T cells and lipid antigens to iNKT cells and modulate their differentiation and function. B cells are also important cytokine-producing cells, which, similar to T cells, can be classified into functionally distinct effector cell subsets based on the cytokines they produce. So, whereas B effector cells produce pro-inflammatory cytokines such as IL-6 and TNF-alpha, regulatory B cells produce anti-inflammatory cytokines such as IL-10 and mediate immune suppression. A lot of my work has focused on studying the balance or imbalance between these subsets and their contributions to various disease settings. So why COPD? So COPD is a chronic inflammatory lung disease that is characterized by emphysema or alveolar disruption, chronic obstructive bronchiolitis, and mucus hypersecretion, which ultimately result in airflow obstruction and breathing difficulties in patients. COPD is in fact the third leading cause of death worldwide, with no currently available treatments to prevent progression of disease, thereby highlighting a major unmet clinical need in the field for better disease management. With regards to B cells in COPD, what we know at the moment is that there are increased numbers of B cells and lymphoid follicles in the COPD lung, and that these associate with disease severity, particularly with the emphysema phenotype. Importantly, there have been studies showing that mice lacking B cells develop a significantly less severe emphysema, thereby highlighting the importance of B cells in driving COPD pathogenesis. If you could see the data here, you'll be able to see quite nicely that mice lacking B cells don't have immune cell infiltrates and they have intact lung structure. So despite the fact that these studies have been published nearly 8 or 10 years ago, there has been very little done in terms of characterizing B-cell responses in COPD patients. And so therefore, we set out to study whether and how B-cell responses vary between COPD patients and controls. And to do this, we used healthy margins from lung resections and matched blood samples from individuals undergoing cardiothoracic surgery. We then isolated the pulmonary mononuclear cells and matched PBMCs and froze them down for subsequent analysis to characterize the different B-cell responses. Similar to previous reports, we also observed large CD20 positive B-cell aggregates which accumulate and persist by the small and large airways in the COPD lung, but not in the controls, as you can see here. So here the CD20 positive B-cells are represented in red. When we then look at the other immune cell subsets, we see that the CD20 positive B cells and the CD3 positive T cells here represented in blue have distinct areas highly characteristic of lymphoid follicles, and that they appear to be in close proximity with CD68 positive macrophages, suggesting likely interactions between these cell types in the COPD lung. We then set up an extensive site-off panel to characterize the different B-cell subsets in matched blood and lung samples of COPD patients and controls. And what you can appreciate here is that when we look at the blood B-cell profile, although there are modest differences between COPD patients and controls, the majority of the differences are indeed at the tissue site. So the profile of the lung B-cells between COPD patients are completely distinct from that of the controls. When we then go on to quantify the B-cell subsets by flow cytometry, we observed that there was indeed an expansion of B cells, BLIMP1 positive plasma cells, and particularly the IgG isotype in the COPD lung compared to the control lung. So in these graphs, the lung samples are represented in gray, the blood in pink, and the close triangles represent current smokers. And the reason we highlight them is because cigarette smoking is a major risk factor for COPD, and so we were interested to understand its contribution to the B-cell responses we see. Rather unexpectedly, we also observed a dramatic expansion of TIM1 positive B cells in the COPD lung compared to the control lung. Now, TIM1 is a phosphatidylserine receptor that is also considered a marker of immunosuppressive regulatory B cells. So as I mentioned, Bregs exhibit immune suppression primarily via IL-10. So we then stimulated the B cells from the COPD lung and control lung with either TLR9 agonist, CD40 ligand, or both, and found that in all conditions, B cells from the COPD lung produced significantly more IL-10 compared to the control lung, suggesting that these were in fact regulatory B cells. Now this was rather confusing to us because, as I mentioned, Bregs via the production of IL-10 can suppress a variety of different effector cell responses, including Th1, Th17, and CD8 positive T cell responses. In addition, B cells, upon interacting with CD4 positive T cells, can induce their differentiation into FOXP3 positive Tregs or IL-10 secreting TR1 cells. So it was rather confusing that we saw this expansion of these immunosuppressive regulatory B cells within the COPD lung and specifically associated with disease severity. However, upon looking into the literature, there is evidence implicating a role for IL-10 in tissue-specific inflammation. So this study by Elias and colleagues shows that the transgenic overexpression of IL-10 specifically in the lung over a period of about 2 to 3 months can cause mucus metaplasia, B and T cell-driven tissue inflammation, and airway remodeling via both IL-13 dependent and independent pathways. So suggesting that perhaps in this context, these IL-10 secreting regulatory B cells may be pathogenic and contributing to tissue fibrosis and inflammation and disease. Next, in order to identify novel pathways that might explain these altered B cell responses in the COPD lung compared to the control lung, we used single-cell RNA sequencing. Our data analysis reveals the presence of 6 unique subsets of B cells within the lung. And when we look into characterize them further based on the known gene expressions, we see that plasma cells are cluster 1 expressing high level of PRDM1 and class switched antibody, cluster 2 is memory B cells, cluster 3 is plasmablasts, 4 is double negative memory B cells, 5 is naive B cells expressing high level of IgM and IgD, and cluster 6 is this population of activated B cells that are uniquely expanded in the COPD lung and express high levels of CD1D. We then looked at the top differentially expressed genes between COPD patients and controls and across the 6 different B cell subsets we've identified. And amongst a number of interesting genes that we see come up, including MMP19, ApoE, etc., we found that IL-10 was also one of the top genes, and that it was specifically expressed in cluster 1 and cluster 6 in COPD patients, which is the plasma cell cluster and the activated B cell cluster. Next, we ran our data through Ingenuity pathway analysis to identify potential transcription factors that might be contributing to this altered gene expression we see in B cells from the COPD lung, and also to understand how the different genes we observe co-regulate one another. And as you can see here, one of the top hits from our analysis was the HIF1 alpha, or the hypoxia-inducible factor 1 alpha pathway. So HIF1 alpha here upregulates PDPK1 expression, which is important for B cell survival, proliferation, and differentiation, as well as ApoE expression, which promotes lipid antigen presentation by B cells to iNKT cells and modulates their function. HIF1 alpha, both indirectly via IL-15 and directly, can also upregulate the production of IL-10 by B cells and therefore expand this population of regulatory cells. So to summarize what I've shown you with a proposed model, what we think happens is that in a healthy individual, the presence of these Bregs is likely to dampen inflammation in response to constant environmental triggers that we're exposed to on a daily basis, but that in genetically susceptible individuals, chronic exposure to cigarette smoke or other environmental pollutants results in increased HIF1 alpha signaling, which then promotes the expansion of TIM1 positive IL-10 secreting regulatory B cells. These B-regs, in turn, can, while they can inhibit Th1 and Th17 responses, can also promote Th2 responses and IL-13 production, as well as activate TIM4 positive macrophages and promote their production of MMP12. B cells, via increased expression of CD1D and ApoE, can also modulate iNKT cell responses by promoting their proliferation and interferon gamma production by these cells, and therefore both directly and indirectly promote tissue inflammation, mucus hypersecretion, and airway remodeling in COPD. So as part of our ongoing work, we're interested to understand how B-cell responses associate with disease severity in COPD patients. And to do this, we're utilizing a longitudinal cohort of patients where we can compare B-cell responses during acute exacerbations and stable disease within the same individual. We are also interested to see how the interactions with the lung microenvironment influence B-cell function and disease outcomes, and particularly how these TIM1 positive IL-10 secreting B cells modulate immunopathology and disease outcomes using mouse models of COPD. I haven't had time to show you the data, but Tracy did mention it briefly. As part of the CIRCLE study, we're also studying lymphocyte responses at up to 12 months of convalescence in long COVID patients and trying to identify unique signatures that associate with specific long COVID symptoms. Finally, we are also studying SARS-CoV-2 vaccine responses, specifically to understand whether the booster doses, the third versus the fourth dose, enhance protective immunity in immunocompromised individuals, specifically those undergoing renal transplant or kidney dialysis. So this brings me to my final and most important slide, thanking the wonderful people I work with. So majority of the work I've shown you from the COPD study was generated by Halima Shua, a PhD student working with Tracy and myself. Beth Potts is a research tech in the lab who has taken over the long COVID work and is characterizing lymphocyte responses within our patient cohort. And Katie Dodd studies B cells in autoimmune diseases. I have to thank Tracy for being a wonderful mentor and also a collaborator on this project, and Sean Knight for being instrumental in recruiting all the patient cohorts that we use to study these diseases. Finally, thank you all for listening. I'm happy to take any questions.
Thanks a lot, Madhvi. Any questions for Madhvi? I'll start off with my lack of B-cell knowledge. So B-regs, have people managed to kind of sort a B-reg based on IL-10 expression? And is there a heterogeneity within that population? Do they make different types of antibody to a more non-B-reg B-cell?
Yeah, so unlike T cells where you have lineage-driven subsets, with B cells, they're more plastic. So depending on the environmental stimuli, virtually any subset can become a B-reg. But specifically, transitional B cells are thought to be the precursors to regulatory B cells. There isn't people have looked for many years, but there isn't a transcription factor that's specifically been associated with IL-10 production. But TIM1 is probably the closest to it. So it's a surface marker that pretty much identifies all subsets of IL-10 producing B cells.
Just seeing those lovely histology images, so do you see any co-localization of your B cells, those B-reg cells, with less regions of fibrosis, or alternatively association of other cell types that might promote fibrosis with those areas?
Yeah, that's a great question, and that's actually what we're trying to do at the moment. We haven't been able to optimize the IL-10 antibodies, so we're working on that to be able to definitively say those are the regulatory cells. But what we have seen is an expansion of FOXP3 positive CD4 T cells within the lymphoid follicles that I showed you, so maybe they're T follicular regulatory cells. Would be quite interesting to look at the interactions once we can uniquely identify our B-reg population.
Thanks, Madhvi. That's really interesting in respect of the COPD in particular. So COPD, many patients will have recurring localized pulmonary infections throughout their disease progression, which clearly will have an influence on the sort of local pulmonary immune response over time. Are you sort of looking into that? Can that be explored in the animal models, or can you explore that in the patients?
Yes, so that's what we're planning to do using the mouse models of disease. So we plan to give these, there's this particular mouse model that induces COPD, and then we plan to expose them to rhinovirus to see how they respond and how those pathways might be associated with these particular TIM1 positive IL-10 secreting cells and how they may be contributing to increased infection susceptibility. So we do think that the presence of these Bregs does promote acute exacerbations and associate with increased susceptibility to infections.